Starting recording in 3, 2, 1, go. Alright, we got activity here. So, speaking of activity, there you are, seated, standing, staring across the cell membrane, crazy structures all around you, shapes and shifting things, yada 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 yada. How do you actually get inside the cell? How do you, or any small molecule, get inside the cell? Now this is important because the cell is of course just the basic building block of all life, so if you can get inside the cell, you can get inside life, you can get inside anything. This has both upsides and downsides, of course. It means that good things can get delivered inside of your cells to do good stuff for you, but it also means that bad things get delivered inside your cells, like viruses, stay tuned, and that's obviously really bad. So, how does this stuff get into your cell? There's a number of ways, but basically it all just comes down to active versus passive transport. Active versus passive transport. There's a number of different styles of passive transport. The names are very straightforward. Active transport basically implies that something is grabbing something on the outside of the cell and then dragging it across the cell membrane and then turning around and spitting it out on the other side. And this is practically speaking how these things seem to work, though it's often more like sort of a twisting. You get sort of a tube-shaped membrane that then twists, and that twisting motion sort of sucks a sort of roughly cylindrical, or roughly spherical, molecule through the membrane and then out the other side. Passive transport is, of course, a passive, and it happens in a couple different ways, but we're going to finish our active transport diagram before I get too excited. So first, drawing my basic phospholipid membrane. You always draw them something like this. Stay tuned, you'll find out why in the end. It's beautiful. I love it. It's the phospholipid membrane. Get used to drawing it. It's one of your best guys. This is a big old channel protein here. There's a couple ways in which these things can go down, but for example, if we have some sort of signal protein over here, and this signal protein is got some mechanism on it that indicates whether or not the internal cell inside the cell it actually wants, say, calcium or something inside of it. It sends then a signal to the outside of the cell, maybe just like a lever switches up, and then something that delivers calcium, or just a calcium molecule itself, shows up and sees that the signal is there and is all like, oh, yep, I want to go inside, and then wanders over towards the membrane protein, at which point either the actual molecule itself, because these are all chemically active objects, triggers a change inside of the tunnel itself, or there's some sort of peripheral molecule required that will trigger that change as well. That's, to be honest, most of the different ways in which these things can happen. This thing then gets put inside here, which undergoes some sort of physical change, which ejects it out the other side. The great advantage to active transport is, of course, you have incredibly selective control over exactly what's going on. One of the most important active transport membrane proteins that we've discovered is, of course, the sodium-potassium pumps involved in regulating the membrane potential of neurons in your brain, which a lot of you out there, of course, already know lots about. So passive transport is also really cool. The first sort of passive transport is one of the simplest and most enjoyable ones, and it's just called diffusion. I love diffusion. Diffusion is really nifty. Diffusion's super basic. For certain sorts of particles, they will simply, they're not that bothered by the cell membrane, and they will simply just move through it all by themselves. Some of them, much more interesting ones actually, will go through the cell membrane, but only if there's particular conditions involved. And there's different sorts of drugs and things that obviously take great advantage of this. The classic example, I think, is probably steroids, which are fat-based and therefore, we'll figure this all out in detail later, therefore will just diffuse quite naturally through the cell membrane and straight into your cells, where obviously steroids can have an immense effect upon you. There are all sorts of natural steroids that exist in your body. Of course, this isn't just the external injected ones, but both of them use basically the same set of pathways in order to get their drug action activated. So that's your diffusion, but then you can also, you can sort of help diffusion in a couple different ways, if that's the right sort of way of looking at it. There are, for example, particles that will diffuse straight through the membrane, but there's also particles that don't want to diffuse straight through the membrane, but will diffuse into the cell if there's a large enough channel available to them. And the simplest way to think of this is if we just have a big enough particle, and it's just a little too large for it to make it through the little gaps in between the phospholipid bilayer, but it is big enough that if you just put a big honking channel protein there, everything sorts itself out and it'll just zoom straight on through that for you. Now here again, I'm drawing some more beautiful phospholipid membrane here, and this is our bigger molecule, and here it is going straight on through this good old channel. Another thing, if we want selective transport, again, we can use sort of a gated system, whereby, what's the best way to do this? I think probably kind of like one of those doors, and some of them are actually shaped like this. A rotating door. So you go in, obviously it should be, it should be like that. You go in and then it's sort of, you can sort of push it around if the key is unlocked, if there's enough of a particular molecule. There's different ones and different structures and way that you can do it. But you can see again, mostly you have a diffusive influence moving particles towards it, and then you have some sort of mechanism that can take over from there and affect the diffusion actually into the cell. So those are some really basic structures that we can use to get in the cell. If I were to choose, of course, I would casually, and we're continuing our little journey where we wander deeper and deeper inside of the cell, I would, of course, choose to take a passive transport. I would wander over here, I think, and wander through the larger molecule. This is, of course, one of the other aspects of these large channel proteins. They are not selective usually, they're not selective usually, and therefore they can allow all sorts of other particles that perhaps normally wouldn't be able to get in. So this means that you can use them in tandem with a bunch of other systems in order to try and limit that, but also there's obvious sort of pros and cons for the body using different proteins in different channels in different places. In practical circumstances, the body uses all of these different things in constant coordination with each other in order to essentially carry out all of the basic different activities that are required chemically in order to keep your body alive, and do a whole bunch of other interesting things besides that. So once we actually make it inside, once we make it inside the cell, we see microtubules. That's the most likely thing, at least. We witness the cell's internal structure. So the cell is not typically just sort of hanging out. It's actually propped up by some number of internal structures. And one of the most common ones in terms of actually creating shape inside of the cell and creating sort of pathways that we can travel from place to place, because obviously we have distinctive things like a nucleus and things inside here, is microtubules. Example, microtubules. Microtubules are essentially just little protein sheets, coiled so that you can do any number of things inside of the cell. They're incredibly flexible, incredibly useful. So, we're still obviously surrounded by liquid. Surrounded by liquid. And that liquid is called cytosol. And cytosol is in the cytoplasm. So we are located in the cytoplasm. The cytoplasm is basically just the name for the inside of the cell. Intracellular space, extracellular space, both totally acceptable as well, of course. Now, the cytoplasm and the cytosol contain all sorts of really important things. The cytoplasm itself obviously has a cytoplasm. It contains all the organelles and vital molecules for cellular function, which is really good, obviously. And then the cytosol It contains the actual fluid dissolvable molecules that your bodies, your cells, sorry, machines require to survive. So that's the basic split between the cytosol and the cytoplasm. Combining those two things, you're now see yourself sort of floating in this weird space that's sort of crossed with this internal structure and machinery. And you can also make out, again, a whole bunch of like big different sections inside of it, of these big different like regions where there's feverish activity occurring. And that activity is mostly sort of consolidated around the center of the cell. So cells are very much a three-dimensional central space where a whole bunch of things are occurring in the middle, a whole bunch of things are occurring on the peripheries, and then you have these different sort of structures in between them serving as important bridges, important focal points for all sorts of different really important processes. So now that we've talked about all these different processes, it seems really important to establish a really basic human cell anatomy. Now, this is not just any old human cell, this is also any old animal cell as well. And we're not going to get too crazy specific with it in this particular moment. There's a lot of different ways to draw these. I think we're just going to go with this one. I'll do it planet style, you're welcome. Here we have the nucleus of the cell kicking in. The nucleoli in the middle. Around the nucleus, we have this nice, interesting structure, which I'm going to try and draw like this. It's the endoplasmic reticulum. We'll see later that basically all that's going on with DNA and the nucleus and all this stuff is information is being stored and then used to generate proteins. And it makes a lot of sense that that information would be stored in the nucleus at the center of the cell, and then it's used to make proteins, or quite a lot of it, just right outside of it in the endoplasmic reticulum. Which makes a lot of sense as well. So there's two real sections to the endoplasmic reticulum. There's the rough endoplasmic reticulum. Reticulum, like so. This is identifiable by the ribosomes on the surface, which make it sort of seem spotty. And then, of course, there's smooth endoplasmic reticulum, as well. Does not have the ribosomes. Smooth endoplasmic reticulum. On the outside, obviously, we have the cell membrane, which we also know as the phospholipid bilayer. That's important. We'll keep coming back to it. It's not going to get old. Microtubules. Can draw some example microtubules in here. Obviously, it's pretty tricky to fit everything into the whole cell, but what else is of value here? Vacuoles. Vacuoles are storage containers for all sorts of different things in the cell. Vacuoles are essentially subsections of the cell wall itself, wrapped into little spheres. It's one of the reasons why the phospholipid membrane is so important and so wonderful to understand properly. Obviously, another huge candidate here, and these guys, I like to draw them as like coffee beans. It's the mitochondria. Give this cell several mitochondria. Mitochondria are positioned often basically just anywhere the cell needs a constant supply of energy. Great examples of this are of course, the mitochondria are often near the outsides of the cell so that they can provide fuel for all the different sorts of receptors that we've talked about. Many of these channels, any of them that require active components, any sort of activation or deactivation or actively moving the thing through, all of those things require mitochondria All of those things require energy, and so one of the simple ways that the body solves that problem is just to move mitochondria directly to that location. The Golgi complex I should put beside a vacuole because the Golgi complex plays a nice prominent role in folding vacuoles. Vacuoles are sort of like the body's own sort of messenger and storage system. You can pack things up and move them around via a number of Golgi apparatus by a number of mechanisms. This is, I think, almost everything in the basic cell that I really want to point out to you. You got your ribosomes, you got your smooth and rough endoplasmic reticulum. There's lots more to it. I suppose we could label the cytosol. It's hard to extract the cytosol from the cytoplasm, but cytoplasm. And I think one last obvious thing is anything outside here is extracellular space, right? So that gives us a way of indicating what's inside of the cell, what's on one side of the phospholipid membrane, and what's on the other side. Because your body is really like this combination of different cells sort of relying on other chunks of cell connected to each other by chunks of protein that the cells themselves generate and sort of exude around them, you get this, you constantly have this important relationship between inside of the cell and outside of the cell and the interactions between them. And in fact, that gradient, that barrier that exists between the two is incredibly important in terms of all sorts of actual functional changes inside of the cell. All right, we're almost done. But it seems an awful shame to talk about the cell, to talk about DNA, to talk about all of this stuff, without also talking about meiosis and mitosis. Meiosis and mitosis. So these are the two types of reproduction. You can subdivide reproduction in a number of different ways, but these are two really important prominent biological forms of reproduction you're undoubtedly familiar with in at least some sort of context. Meet the mushroom. The mushroom. The mushroom. I'm excited to meet the mushroom. You're also excited to meet the mushroom. There's the mushroom. Ah, Mr. Mushroom. Now, you, of course, already know the mushroom well. This is the fruiting body. The fruiting body of a fungus. And the fungus you can think of as consisting of two general parts. It has fruiting bodies, and then it also has this complicated root-like structure, which is really the vast majority of the plant and what is the most important part of it. It's not a plant, sorry, the fungus, of the organism, and the most important part of it. This is the mycelium. Mycelium. Spelled correctly. He lied. Spelled incorrectly, one L, sorry, mycelium. So this is the mycelium. The mycelium also has these little tips coming off of it called hyphae. But to be honest, this is the much more important word for this particular moment. So the reason we are interested in the mycelium is because the mycelium can reproduce sexually or asexually, depending on environmental conditions. Sexually or asexually. Now, I gotta go double check so that I don't mess this up, but I'm pretty sure, definition, that mitosis is asexual reproduction. And I'm also pretty sure, definition, that meiosis is sexual reproduction. What does this mean? So, if we are to look at a little chunk of mycelium of this sort of mushroom root, then it has two different available routes to it. It can either go the mitosis route, whereby it directly releases spores, and these spores are basically just clones. In fact, they are just clones of the original mycelium. Or it can take what's typically a more demanding route. Ooh, there we go. It can produce an entirely different set of cell. It can basically take its root sections and create a new, different copy. Heterokaryotic stage. And then it can perform some sort of sexual process. We need a zygote here. To produce its spores. And this gets you... Get too carried away. This gets you a bunch of spores that are genetically diverse, as opposed to a bunch of spores that are clones of the original mycelium. Genetically diverse mycelium. As a general rule, then, this is the sort of activity, the mitosis versus meiosis loop. The mitosis loop is typically seen as less demanding on an organism's resources, whereas meiosis is more demanding. However, meiosis provides a whole bunch of protections against genetic predators in particular. The mitosis really does not. If you have a particular disease that attacks a particular genetic feature, mitosis requires all of the mycelium that produced from that particular process to be affected by that same disease, whereas meiosis gives an opportunity for that allele to just disappear out of a generation. We'll start to look a little deeper into how that all actually functions in the future, and yeah, look these things up. They're great. You'll break them all down in your biology careers, I'm sure.